So, as we started yesterday, we are going to look at the protein purification or purification of proteins. So, you know that generally proteins are found in a cell, and for you to get that proteins out of a cell, you have to apply a lot of techniques. There's a lot of a series of isolating techniques that we have to apply when trying to isolate proteins from a cell. So actually, in a cell, we have a millions of proteins. We have a millions of proteins. So if you want to study a particular
Ya shiga
proteins,
tare da
the
Dr.
best
MK.
thing,
Kin
the
san kin ce
best
wurin
thing,
yin place it.
the best thing if you want to study a particular protein is to isolate it from the other proteins. It's like, for example, if you want to toss a girl, the best thing is to isolate her and then you toss her. So that is the best thing. And also in the case of proteins, if you want to study a particular proteins, so you have to isolate it. You have to extract it from the other proteins. So that is what we are going to look at in this lecture. How are we going to isolate the proteins from cocktails of proteins? So now, why do we need to purify proteins? We need to understand why do we have to purify proteins? So number one, to identify the structure and functions of a protein of interest. Sometimes if you have a proteins in a cell, because there are so many proteins there in that particular cells, you may not probably know the functions of individual proteins that are present in that particular cell. So, but the best thing if you want to do it is actually you isolate the proteins. So once you isolate the proteins, you will be able to know the structure and the functions of the proteins. It's simple. Because when you said the structure, it's different. Remember, yesterday we talked about there is primary, secondary, tertiary, and quaternary structure of the proteins. So, if you want to understand the structure and the functions of a particular proteins, you have to isolate it. So that is one of the reasons why you need to purify proteins. For you to know its structure and the functions that it elicits. We have some proteins that have that are linear, some they are twisted, and there are some proteins have different functions. So maybe you have a target proteins and you want to know how that proteins look like and the functions of that protein. So you have to isolate it. That is one reason of purification of proteins. And second, to study protein regulation and protein-protein interactions. For example, now, if you isolate a protein, if you isolate a proteins, you can be, or you'll be able to know the protein regulation. That is what it control, what the activities it does in that particular cell. Does it actually control a particular pathway? Because in each and every, remember that from our previous classes, we talk about metabolism. And this metabolism is divided into catabolism and anabolism. And there's a different pathways. So in each and every pathway, example of pathway, we have like glycolysis. So you can actually be able to understand the protein regulator. That is, which of the proteins is responsible for regulating a particular pathway. So if you are able to isolate a particular proteins, you can be able to know whether it is the control point of that particular protein. So also at the same time, you'll also be able to know the protein-protein interactions. That is, after you isolate, you will be able to know whether the proteins that you are isolating is interacting with other proteins for it actually to carry its function. So protein purification will be able to address which of the proteins is responsible for regulating or for controlling a particular pathway. And so also at the same time, you will be able to know whether the proteins is interacting with other proteins for it to carry out its acts. And then the next thing is to produce antibodies. Generally, some of the proteins that are found on some of the pathogens or some of the biomolecules that are found on some pathogens, there are proteins. Like for example, we have what you call spike proteins on coronavirus. So since we have spike protein on coronavirus, using this purification process, you can be able to actually get the virus, isolate the proteins, which is the spike proteins on that coronavirus, and actually study its structure, and then from there you will be able to develop an antibodies that will actually fight that particular spike proteins. And by doing that, it means that you end up of producing vaccine. And so also at the same time, you can isolate proteins for you to actually use that particular protein in access. You can access. That is when we said assays or proteins is assays or enzymes assay, it means that maybe you want to know how that enzyme is able to convert a particular substrate to product. And then, of course, if you want to carry out a structural analysis by X-ray crystallography, you also have to isolate proteins. Remember, X-ray crystallography is like a technique that uses the principle of diffraction method for you to actually be able to understand how the structure of a particular proteins look like in terms of the atomic arrangement, the space, and so many things. So these are the reasons why we actually isolate proteins. So you should know, one, to identify the structure and the functions of the proteins. Second, to actually study protein regulation as well as the protein-protein interaction. Then you can also study proteins for you to raise or to produce antibodies against those proteins. And you can also isolate proteins to assay or to use it in assaying. And then for the structural analysis using X-ray crystallography, you also have to isolate proteins. Okay, then the next thing that you are going to look at is, where do we get these proteins? What are the sources of these proteins that we are talking about? Although actually from the previous class, I also told you how these proteins are found. So these proteins are usually found in the frame fat of organisms or even in the whole organisms. So you can get protein from whole organisms, you can get protein from organs, you can get proteins from tissue, you can get proteins from embryos, you can get it from tissue culture cells, you can even decide to get a gene, a particular gene, and actually use expression system to express the protein. So it means that you can actually express the proteins and you can also get it from there and manipulate it. So now, ladies and gentlemen, why do we have to know this? Number one, if you have a particular enzymes that you are intended to target, first you have to check from the literature. Where is that enzyme is located? In which organism is it located? Is it in microorganisms? Is it in human being? Is it in bacteria? Is it in fungi? Or is it in virus? So you need to check the literature so that at least you will have an idea of which organisms is having your protein of interest. Assuming now you want to actually get spike proteins. And of course, you know that the spike proteins is the proteins that usually found on coronavirus. So you can't just say, okay, you want to go to the liver tissue and get spike protein. It's not possible. So that is why you need a proper so you need to actually set your target. You need to actually know your target. If, like, for example, you want to get some proteins and maybe it is already known or it has been established that that protein is present in the mitochondria of the liver cell. And you now decide to say, okay, you want to go to the brain cells the and get the cells from the brain and start thinking to get that enzyme. So it's not possible and it's not advisable. So always, if you want to isolate a particular enzyme, try to identify the source of that particular proteins. And now, for example, let me just say this. Assuming now you have a similar proteins, or maybe you want to get protein A, and that protein A is present in human and it's also present in microorganisms. So which one do you have to do? Are you going to get it from the human or you are going to get it from the microorganisms? I'm asking question, please. Who can answer this? You want to get protein A. And that protein A, it has been reported from the literature that you can get it from microorganisms and you can get it from human. So which one are you going for? Are you going to get it from the human or from the microorganisms?
Microorganism.
Microorganisms.
Microorganisms.
Yeah, the microorganisms is the best because if you say that you want to get it from human, so you have to slaughter that particular human. So also not even human or alone, even in other animals, even if it is there in other animals, because you have to slaughter that animal and get the liver from them or and get the organs where the enzymes is being localized. So but using microorganisms, you can easily manipulate them, you can easily culture them, you can easily regenerate them. So it is easier to get that particular enzymes from microorganisms. So that is why it is advisable when you have a particular enzymes that you are targeting, you have to isolate, you have to try as much as possible to identify the source. And sometimes you see, as you said, you can get from the whole organisms, organs, tissue, embryo, and also tissue culture cells, as well as the proteins from the expression system. So you see, when you said expression system, it means that what you do, although I think we did not shout a lot on the concept of frontal dogma of molecular biology, but we have DNA, and in DNA we have genes, and each gene is responsible for producing a particular set of proteins. So now, if you want to get a particular proteins, you can just decide to go and check in the from the DNA of that particular organisms. You can get the genes. So if you can get the genes, you can easily apply the knowledge of genetics engineering and biotechnology to use that genes, then you get the proteins. And that is what we mean protein expression or proteins from the expression system. So sometimes we do this. Like for example, currently we are running one particular research. Generally, we know that mosquitoes is responsible for transmitting malaria and some other infectious diseases. And of course, from the literature, we know that generally insects, they are arthropods, so meaning that they have chitin. So we actually trying to develop a particular insecticide that is a compound that can be killing the mosquito because there is a problem of resistance on the mosquitoes. There are so many insecticides now that they are inactive because the mosquitoes have become resistant to them. So as a result of that, we are now thinking of developing new insecticide. So we now be able to remember, okay, so they have chitin, and then when we are checking from the literature, we were able to come across entomopathogenic fungi. That is some fungi that can easily kill mosquito. So as we are checking from the literature and we were able to identify that those entomopathogenic fungi, they are producing an enzyme which is chitinase. So chitinase is an enzyme that can easily break down and chop off the chitin. So as a result of that, some of those microorganisms, because they have this chitinase, then they can easily kill the mosquito, and it has been established. Okay, then when we actually see that this thing has already been established, then now we now think of, okay, what is the best way? What is the best way to produce this chitinase? Then we now say, okay, so the best thing, since these microorganisms or this entomopathogenic fungi, they are producing chitinase, of course, it means that they have the gene in them that is responsible for producing that chitinase. We said, okay, so let's isolate the gene. Let's just get expression system and express this protein. Then we now use the knowledge of genetics engineering as well as the biotechnology, and we are now able to produce that particular proteins. And interestingly, the proteins was able to kill the mosquito. So it means that using the expression system, so that is how we were able to get that protein. So the best way sometime if you want to get a particular enzymes, the best thing to just is better you apply this expression system to get the proteins because you can easily get it in much more purer form and you can easily work with it. And with this expression system, you can produce as many proteins as you want. But sometimes if you say that you want to use organisms, especially like if the enzymes is from animal source, you have to slaughter so many animals before you are able to get the required amount of the proteins that you are needed. Okay. So this is actually in with regards to the source of the proteins. Okay, so now what are the steps of this protein purification? So now assuming you have an idea, okay, this enzymes is present in a liver cell. So how do you do it? The best thing or the first thing that you are going to do is that if it is a liver cells, then you have to slaughter animal. You have to get the liver. After you get the liver, then you homogenize. After you homogenize, you centrifuge. After you centrifuge, and you lyse the cell. After you lyse the cell, you have to uh you have to after you homogenize, then you have to lyse the cell, because after you homogenize the cells, you are going to get like different component of the cell. So it's going to disaggregate the tissue into individual cells. Then you can now get the individual cells, put it in a suitable isolation media. After I put it in a suitable isolation media, then you open up the cell. After you open up the cell, then every component, every content of that particular cell then will be now released into the suitable isolation media. And then once you do that, then the next thing you do centrifugation. You centrifuge. Then after you do centrifugation, what next? The centrifugation will now separate different component of the cell. Because remember, we have organelles of the cell. We have nucleus, we have mitochondria, lysosomes, ribosomes, Golgi body. So when you do the centrifugation at different revolution per minute or at different gravity, then you will be able to separate the individual organelles of the cell. So assuming now your enzymes is in the mitochondria, then you now apply or you now use the revolution per minute that will actually settle down the mitochondria, and then you remove the supernatant, and then you now have the mitochondria. Then after you have the mitochondria, then you open up the mitochondria. Then you now go for stabilization of the protein. And then after you do the isolation, then after the isolation, then you apply some of the basic techniques. So you see in the first step, you start with the solubilization of the proteins, and this comprises homogenization, centrifugation, and then filtration. Then after filtration is stabilization of the proteins, and then lastly, the isolation of the proteins. And we have some biological material themselves constitute a clear or nearly clearer protein solution suitable for direct application chromatography columns after centrifugation or filtration. So it means that there are some proteins that after you do centrifugation, because they are very clearer, then you can just simply subject them to chromatographical techniques or chromatographic techniques. And these chromatographic techniques include like gel filtration, column chromatography, affinity chromatography, size exclusion chromatography, then depending on the types of the proteins and the techniques that you are going to apply. Okay, but the most important thing that you should understand here is we are interested to isolate the proteins. And this process, it involves a lot of techniques. And at the end, after you apply all these techniques, you will end up of having your proteins. And then from there, you will be able to determine the amino acid sequence. Okay. So now let's start with the homogenization. How did you homogenize? Then after you have your tissue is we have different homogenization techniques. And these homogenization techniques depend on the types of the tissue that you are working with. If you are working with a fragile tissue, you can just use dissociation, freeze and thawing, enzymatic or cleavage of the cells. You can even apply freeze and thawing. But if you want to do it in larger scale, then you apply high pressure homogenization or beat myelin. So you see sonification is just that you are using sound energy open up the membrane of the cell. So also applied in a French press, you just trying to open up the membrane of the cell. And of course, in the case of enzymatic or chemical cleavage, in the case of enzymatic, we have some enzymes like xylinase, we have like xylinase, we have like chitinase itself, we have like proteinase. They are the proteins or enzymes that can be able to open up the membrane of the cell so that the content of the cell will be released into the isolation media. So also Chris and Towing is the same technique. So all these techniques, as you see them, they are techniques that I use because I don't want to go into more deep on this. It's just for you to have a general basis of how we are doing the things. Okay. And then the next one is centrifugation after homogenization. So you homogenize, you open up the cell, you remove the cell membrane, and all the content of the cell is being released into the medium. So the next thing, the centrifugation is done to isolate the proteins that are component of the membrane or subcellular assembly. For example, mitochondrial protein. So if the proteins is in the mitochondria, so the next thing is you have the tissue homogenized after homogenization, you centrifuge as 1000, and at the end you'll have pellets. So in the pellet you have unbroken cells nucleus and chloroplast. And then at 10,000 you will get the pellet mitochondria. At 100,000 you will get mitosome. So it's depend on actually what you are looking for. So it means that if you want to get the mitochondria, then you now centrifuge at 100 at 10,000 times G. Then you get the mitochondria. Then after you get the mitochondria, so you have the cell lysate is centrifuge at a speed that removes only the cell component denser than the desired organelles. So you see, this is how you do it. Then you navigate your desired organelle. And then the next thing after you do that then is now stabilization of the protein. So you have to stabilize the proteins at some, like for example, care must be taken to preserve protein structure and function after it is being removed from its natural environment where it was stable. So remember actually when you have these proteins are in a cell, so they have a particular environment and they have the conditions that make them to be stable. So when you bring them out of that particular cells, you have to make sure that you stabilize that protein so that at least the structure and functions is not being tempered. So as a result, it's not being altered. So it's not being altered. So as a result of that, then you have to apply some techniques that actually will prevent those proteins from being degraded or from being destroyed. And one way of doing that is to actually first understand in the natural environment, is the enzymes in acidic pH? Is it at neutral pH? Is it at basic pH? So if it is at acidic pH, then you have to make sure that you provide an environment where where it is actually acidic so that the enzymes will be stable. And then the next thing is the temperature. Some enzymes actually, so protein purification is normally carried out at very low temperature. So we carry the proteins, whether sometimes we allow it at 0 degrees centigrade or at 4 degrees centigrade when the enzymes is not actually active. So while some proteins are thermally stable at high temperature. And you also have to introduce inhibition of proteases because sometimes if you actually open up the cell after homogenization, you centrifuge, you get the organelles and you open up the cell. At the end, in your suitable media or in your media, you will have some proteases that can easily chop up and break those proteins. So that is why you have to add protease inhibitors. And the retardation of microbes that can destroy proteins. So you can use sodium azides. is in the medium so that at least some microorganisms cannot come across those proteins and destroy them. So this is actually the isolation of proteins. That is the step. So method of isolation of proteins differ from one protein to another. So it might not necessarily that you are going to apply the same techniques. It might not necessarily that you are going to apply the same techniques in isolating different proteins because the structure of the proteins varies. So as a result of that, you look at the proteins itself, you check the literature, how it is structured, it looks like how sorry, the nature of the proteins as well. Then from there, then you apply the the suitable isolation techniques for you to isolate the proteins. And these characteristics that we use, we have based on the solubility of the proteins, you can apply certain out. Based on molecular sizes, then you can use dialysis or gel chromatography. And for the ion exchange, you can use ionic charge, you can use ion exchange, gel electrophoresis, and also electric focusing. And then based on the binding specificity, you can use affinity chromatography so that at least